C, we got operator, promoter, plus one region, and different region. For D, none of them. And for E, we got lac A, lac C. For do it, we got glucose, lactose, and then part B, we got lac B. Now, part B, we got adenyl cyclase. D, we got lac B. E, lactone, lactolide. F, attenuation, and G, varilidector.
All right, so I'm in charge of number 3 today. So, for letter A, it talks about how glucose is absent and X-gal is present. So, regardless of what's going on, even if we have our X-gal, which is our blue substrate, because the IPTG is absent, there's not going to be any, it's not going to be transcribed or anything, so you're going to end up with just normal, and there will be no blue expression. For B, the IPTG is present, glucose is absent, and X-gal is present. So because you have your X-gal and the IPTG, you're going to have the transcription, which is going to eventually allow the blue substrate to be expressed. So you're going to have normal functions through your lac operon. For C, your IPTG is present, glucose is absent, and your X-gal is absent. You're going to have normal because there's no substrate for it to express. There's no blue gene for it to show. For D, it says the IPTG is present, glucose is present, and the X-gal is present. So it says right in here, and just above all the numbers, that the abundant ATP is present in all cases. So because all three are present, we put blue. Reason being is if there's an unlimited amount, it just says abundant, so it may not be exact, but should the glucose be used up before the ATP is, you are going to have a little bit of synthesis for your IPTG, which is going to allow for the blue X-gal to be expressed. For E, it says the IPTG is present, glucose is present, and the X-gal is present. You're going to have normal just because there's no X-gal and your glucose is always used up first before any of the other sugars.
Okay. Alright, so I have 4. If you don't understand me, go and watch her videos. It's kind of a bit, she does diagrams and things, and it helps me a little bit to visualize it. So if you don't understand me, go watch her stuff. Alright, so for number 4, we're trying to decide whether these E. coli strains that were given here display constitutive, meaning that they're always turned on, always producing. Inducible means that they're on and they just kind of work normally. Or non-inducible, meaning they're turned off, they don't work at all. Alright, so for A, everything's wild type, meaning that it's just working normally. So it's going to be inducible because it's going to be a normal working gene. Everything works well. B, we put non-inducible because our, what is it called? Our LACL, that should be L's, I guess, but to me it looks like I's. LACL is defective, or there's none in the actual gene, which means that the repressor can't be removed. So if we had our thing here, this is our repressor preventing transcription. It doesn't have lactose to come and remove it so that it can be transcribed. For C, we put constitutive because our operators actually in both the chromosome and your plasmid are defective, which means that it creates a defective repressor. So your repressor is just the fact that it's going to be on your RNA, but it's floating around. So it's like, yeesh, and it won't bind to the RNA. So the RNA is continuously being used because there's nothing to stop it. D is, we put non-inducible because our LACL is actually defective, or there's an absence of lactose in our plasmid. And even though in our chromosome it's normal, from what we could understand from her logic in her video, the lactose kind of takes precedence over whatever's in the chromosome. So because of that lack of lactose, it's not going to work at all because your repressor is back, just like B. And then E, we put constitutive because again, our operator is defective in our plasmid, which overrides your chromosome, making it so that it's always going to be continually on continuing processing. So yeah.
All right, so we might kind of tag team number 5. I might just get a little long-winded and just do all of it, we'll see. But for A, I really don't want to take a whole lot of time and get into the little descriptions above, so make sure you're actually looking at the little specifications that the assignment has above it. But for letter A, we have a defective LAC-L gene, which if your LAC-L gene is defective, you're not going to have either, whether you add lactose or you subtract lactose from the test tubes. For B, kind of goes along with what we had on number 4. You have that defective operator, which is going to continually allow for that replication. So you're going to have 1, and it's only 1, not 2, because we're only dealing with one level. We're not dealing with the chromosome and the F plasmid. For C, we've got 2 for each. Reason being is you've got normal function for the chromosome, and then you've got a defective operator on the F plasmid, which is just going to allow, the chromosome's going to go till it runs out, and then it's going to go to a certain point, and the F plasmid is just going to continually go until it runs out completely. And so you're going to get levels in both. So you should have 2, regardless if there's lactose or not, if you added it, because there's lactose already present. And for D, you want to do it or you want me to keep going?
Keep going, you're doing great.
All right. So for D, I've got 0 for both. Reason being is that you've got an inoperable LAC-L gene, and then you're going to have, which will automatically wipe out any of the process to get to go through the lactose. For the F plasmid, you've got an inoperable promoter, which automatically means it's non-functioning. There's not anything going to happen. You can't use any of the lactose up because that is a very important process to allow for the processing of that lactose. Then your proteins can't be made and your enzymes can't be made to process the lactose. So you're going to have 0 for each one, regardless if you add it or subtract it from the test tube. E is kind of the same thing, but you're going to have 2 defective LAC-L genes, and so you're not going to be able to. The repressor is not going to attach. You're not going to be able to go through any of the lactose that's provided. So there you go.